Organizers for the kind invitation to this very interesting meeting. And so indeed, so I'm going just to talk to you about the modulation of the gut microbiota by nutrition, and so to try to unravel what is finally the interest, just to focus on this gut microbiota. So what is important to know, and maybe if you just have to remind something, just to keep in mind those two figures that, in my view, represent the gut microbiome. First, it is the iceberg, because honestly, when I started working on the gut microbiota and nutrition, it was more than 20 years ago. The techniques available for the analysis of the gut microbiota were completely different. We could only focus on the microbe that could be cultured. Now, the genome of the bacteria have been sequenced, and it means that we have now access to molecular biology technique to analyze the gut microbiota. And it is, in my view, maybe the reason why now we see more and more studies taking into account the gut microbiota in different pathophysiological conditions. The second thing, it is a gut genus with the two faces. When we have a look to the gut microbiota, so we harbor hundreds of billions of bacteria inside our gut, and so some of those bacteria could be harmful in some cases. And so we also harbor some bacterial components like lipopolysaccharides, for example, and if those lipopolysaccharides go into the blood and translocate through the gut, it means that you will get inflammation. So it means that we have to keep those microbes at bay. And normally, when everything is okay, we do it. And so we share, finally, our life with this ecosystem. The problem is that in some pathologies, and I will only focus on obesity, diabetes, and related metabolic disorders, this ecosystem is disturbed. And so it means that the composition of the gut microbiota, but also the metabolic activity of this gut microbiota, is not the same as compared to the lean individuals or non-diabetic patients. And I don't want to go into detail of the changes of the gut microbiota, but we can see, for example, that there is a decrease of the bacterial diversity associated with the obesity, but also a decrease of some interesting bacteria because of their role for the gut function and for the host function. For example, in obesity and diabetes, there is a drop of some bifidobacteria that play a role as educators of the immune system, but also a drop of some bacteria that play a role in the gut barrier maintenance, like the Akkermansia mucinifila or anti-inflammatory as Faecalibacterium prosnitzi. You don't have to remind those names, but just to remind that finally there is a drop of some very interesting bacteria for our physiology. On the other hand, for sure, there are some bacteria that are gaining interest in view of these disturbances, and there are also an increase of some potentially pathogen bacteria occurring in this case. Then a paper was recently published, because if we observe the dysbiosis, what is the main component? So are there our genes? Do they play a role in the occurrence of the gut microbiome and the maintenance of the gut microbiome? And indeed, it seems that it is mostly the diet which is important in order to maintain and to explain the selectivity of the gut microbiota in a certain condition. So you can see here on these figures and very recent papers that all the blue arrow correspond to some elements linked to the diet that are responsible for shaping the gut microbiota. Then the microbiota really depends on our nutrition. If there is a dysbiosis, and if the nutrition plays a role, so can we think about having some dietary advices to try to improve the gut microbiota and thereby to have an effect on health? There are several methodologies or ways to modulate the gut microbiota, and on these figures you can see that there are some fecal material transfer that are proposed, but also probiotics interventions, meaning that you can give some bacteria to the people and to try to implement those inside the gut. But I will talk to you about nutrition. And the nutrition means that there are some nutrients that mostly escape the digestion in the upper part of the gut. They can meet the microbiota, they can meet the microbes and be used by those microbes, and thereby they can modulate the gut microbiota. So The story started, I would say, quite a long time ago. Now, it was more than 15 years ago, with a compound that has been isolated from chicory root. Why chicory root? Because at that time, the sugar beet was not really well seen, well perceived, because there was a drop of the sugar intake that was really put forward. And so the techniques that were used for extraction of sugars from the sugar beet could be also used for the extraction of some dietary compounds like inulin from the chicory root. And so the inulin, which is the compound that can be extracted, it is a dietary fibers, and due to the chemical structure you see here, it is completely resistant to the digestion by our own enzyme. But some bacteria are expressing enzyme and are able to use those nutrients as a substrate. So inulin can be used and modified by the bifidobacteria. They use it as energy substrate and they acquire proliferative advantage. So it means that when you eat these type of compounds, inulin, you boost some bacteria like bifidobacteria. But we know now that it is not only the bifidobacteria that take advantage, but it changes also the ecosystem and can modulate other types of microbes. Then, what is the effect of those changes of the gut microbiome by inulin on the host health? I will just show you one slide on preclinical studies in animal studies showing that finally, I would say quite logically, the story starts in the gut. When the animals, obese animals, for example, are eating the inulin-type fructans, so it modulates the gut microbiota, but it also modulates very important key gut functions. And I will concentrate on one example. So when the animals eat these type of fibers, finally these type of compounds, prebiotics, it increases the number of endocrine L cells in the gut. And thereby, you produce these hormones like glucagon-like peptide 1, GLP-1, that may play a role on the control of satiety or glycemia. So it means that for sure those changes may have an effect on the gut and thereby explaining systemic effect that could be important in the context of obesity. But we are not here to treat mice, and so it's important just to talk about human studies. And so I will show you briefly 2 slides showing that indeed this hypothesis of the change of the gut microbiota associated with key gut functions is also true in humans. Here it is a randomized double-blind parallel placebo-controlled studies performed in healthy volunteers that have been fed either with the short-chain inulin, 16 grams per day, or a placebo, which is a maltodextrin. And as you can see on the left-hand side of the figure, is that when the subjects after 2 weeks are subjected to an oral glucose tolerance test, indeed the glycemic response is lower in the individuals who had received the inulin in their diet before. And as you can see on the right-hand side, it is linked to an increase of the secretion of these hormones, glucagon-like peptide 1. So it means that some mechanisms that have been shown in animals can also be relevant in humans, and it may explain the hypoglycemic effect of this type of treatment. We have performed a very small study, intervention study in obese individuals. It was a double-blind placebo-controlled intervention study once again, but longer, 3 months in obese individuals. And what I would like just to show you as a message here, it is that when you give the inulin to the obese individuals, you boost the bifidobacteria, but not all the bifids. And so when we have a look to the types of bifidobacteria that are increased, some of them are clearly correlated with a decrease of the adiposity in the obese individuals. And you can also see some change of other bacteria, like Akkermansia mucinifila, that are involved in the gut barrier function. So we don't eat chicory root. And if you do, please send me the recipe. So it's very important to know what we have to eat in order just to get enough inulin, and if those foods are able also to have a role in nutrition and health in this context. So we have recently performed a study. It was once again in healthy volunteers, and so they were quite young and healthy, and so because they were recruited at the university, near to the university hospital and university restaurant. I will tell you why. And so there are the exclusion criteria that are presented on this slide. And you have to know that in these protocols, so the individuals arrived for the beginning of the study, and so they were given during 2 weeks a very controlled diet, quite rich in inulin. So we had sampling before starting this protocol, sampling after the 2 weeks of intervention, and they came to the university restaurant every day, received their meal for the evening and so on. It was really well controlled. And thereafter, they could come back to their usual diet. So we controlled by questionnaire, but they came back to their usual diet, and we took samples once again after 3 weeks. What are the data? What do you have to eat if you want just to have inulin? You have here some examples. So in view of analytical measures, so there are preparation with artichoke, with Jerusalem artichoke, with onion soup or puree, salsify, and so on. So we have now established quite a lot of recipes containing this. And the table below just shows you the amount of fibers and of inulin that you can have before, after coming back to the usual diet. There are no changes of the macronutrients during this protocol, but you can see that for sure, as expected, we boost the dietary fiber intake and in favor of inulin as major fiber, because the amount of fibers is almost doubled. But we can see also that thereafter, after the 2 weeks, when they can come back home and eat as they want, so there with a drop, and it comes back to the basal value of the dietary fiber. What we did in these experiments, so at each test day, at time 0, 1, and 2, so the volunteers came in the lab, which is a clinical lab, and so they were fasted, and there they were given a load of inulin in order to try to know what is the tolerance towards inulin and what is the fermentability of inulin in the different cases. And so to measure the fermentability of inulin in those patients, so we gave a load of inulin and we measured the expired hydrogen, which is one gas coming from the fermentation of the inulin. And what you can see on the right-hand side, it is that indeed, when we compare the hydrogen excretion at the basal value, you can see that it is mostly the same in the 3 groups, except for some individuals. And those individuals, which are the point on the upper, were the one that had Jerusalem artichoke soup the night before. So it means that you keep the fermentability capacity if you eat those type of compounds just the night before, and so it takes time just to have the effect on the fermentation. No change globally on the post-load hydrogen excretion. What was quite interesting, it is the fact that we had some changes of the behavior. So for sure, we tested for the tolerability and gastrointestinal tolerance of inulin, and it was quite nice to see that finally it was really improved after the dietary intervention. But most interestingly, we also had some food questionnaires just to assess the envy to eat sweet, fat, and also satiety in the individuals. What you can see here is that you have changes in satiety that persist even after coming back to the usual diet, and you had also the same pattern for the sweet envy and salt envy, for example. So by this intervention, you can change the way that people perceive the nutrients. Another thing also important, it is that there were no change in the perceived stress score, because it was subjected by some animal studies, but we also had hedonic attitude for vegetables and some vegetables containing inulin that were quite high before the study and remained stable throughout the intervention. So it means that it is maybe not the relevant for the general population. Maybe those students who decided to be included, they already liked maybe the vegetables. So maybe it is one of the biases of this study. What about the gut microbiota? It is exactly the same type of changes of what I have shown you with the fiber intake. You can see here, if you only have a look to the back of this slide, that as expected, we have an increase of the Bifidobacterium genus, a huge increase after the intervention, and it comes back to the normal value when they come back to their normal diet. But we have also a drop of some bacteria that can be related to gut discomfort. So from this study, what can we say? We can say that inulin-rich vegetables can be well tolerated in those individuals, healthy individuals. They allow significant changes of the gut microbiota as observed with isolated compounds, but it is reversible because of the change of the food habits after. We can also correlate and try to find which bacteria can really be related to the gut tolerance. And so we could point out in this story, for example, that the level of some bacteria, like Prevotella, for example, were really related to the gastrointestinal tolerance versus the fermentation. So what is nice is that we can now think about having an intervention study in obese individuals, taking into account some nutritional advices and dietary advices, and not only with dietary supplement with a compound. And it is what we have done, and I won't show you all the details because it's really, I would say, quite a new story. So we just have unpublished data. I will show you some of them. So we performed an intervention study. It is a multicentric intervention study. So at the beginning, we wanted to recruit 150 patients. And so after the end of this story that was ended in June this year, so we could finally add all the samplings necessary in 101 patients finally. They were split into two groups. One group were treated with inulin, addition of inulin, together with recipes with vegetables quite rich in inulin-type fructans, and the placebo group received the maltodextrin as well as recipes and advices to eat vegetables for sure, but which are not necessarily rich in inulin. And so I won't go into detail, but you can see that we have made a lot of measurements and sampling in those patients, and so the analyses are going on for the moment, but it also allows us to have a first view of the set of data we have obtained. What about some parameters? I will just show you two slides. If we focus, for example, on the changes of the body mass index, and it is on the left-hand side in the placebo group before and after the intervention, and on the green line you have the treated group, inulin-type fructans treated group before and after the intervention. And you can see that in both placebo and treated group, we have an improvement of the body mass index. And we could see also that there are a huge variability in the response towards the treatment. For other parameters, the data are significant only in the inulin pretreated persons. And so, for example, diastolic blood pressure can be influenced by the inulin intervention in this story. But once again, there is a huge inter-individual variation. How to address the potential role of the gut microbiota in the inter-individual response? Finally, can we ask the question, the individuals, they eat normally the same thing, if they are really compliant with the diet, for sure we check for that, but there are some individuals that really respond quite higher to the diet in terms of decrease of body weight or improvement of metabolic disorders, whereas other individuals eating the same thing, they don't respond. Is it the gut microbiota that plays a role in this story? Then we have to go to mice, because we cannot make the transfer of microbiota from one human to the other. Some people are doing that, just in science also, in the diabetic patients, but it is really very difficult to have as application, as trial. So we had mice, and what we did to mice, so we treated them with antibiotics, so we cleaned the intestine, and then we transferred the mice with the gut microbiota coming from the patients before they started their treatment. And then we gave the mice a high-fat diet together or not with the inulin. And we wanted to test the hypothesis, if we transfer the initial gut microbiota from the patients to the mice, do they respond differently to the dietary intervention in order to know is the gut microbiota responsible for the differential response? And it was quite surprising. I just show you once again 2 slides. Here you see the body weight of the mice, and so you can see that as compared to the control mice, the D1 are the mice that have been transferred with the gut microbiota from the patient D1. And you can see that those mice highly respond to the high-fat diet by increasing their body mass index. And the inulin has an effect in these mice, not in the other groups. And once again, when you have a look to what happens in the patients themselves that have been treated for 3 months, it is only the patient 1 in this group that responded to the inulin, dietary inulin intervention by a decreasing in body weight. Then it means that in this case, yes, some features associated with the gut microbiota may explain a differential response in terms of evolution of the body weight. We are not using mice only to have this proof of concept, but to analyze the mechanism. And to make a long story short, we could also see that in these mice of patient 1 transfer, there was an activation of the brown adipose tissue that could explain part of the effect on the BMI. So here we can say that finally the dysbiosis is associated with a lot of pathologies, including obesity, diabetes, and related metabolic disorders. But we can have an idea of which type of food can be proposed in order to modulate the gut microbiota, knowing that maybe not all the people will respond the same way to such a dietary intervention. I won't say that the gut microbiota is explaining everything in terms of individual response, but it could be considered as one of the biological elements to take into account in the differential response. And so there are several studies going on for the moment, and one study in which Martin Laville is collaborator, the fiber tax study, in which we test other types of fibers with the same context of individual response. And I thank you for your attention and all the people in the lab who are working a lot on this. Thank you.